So, chapter 5 stuff. This was the chapter that taught you how to sequence a given amino acid sequence, how to figure out what amino acids are in it. The way to solve this is a step-by-step process, some of which you should know a little bit of information about before going in, and that, for example, is Edmonds reagent. If you're given a clue that tells you Edmonds reagent results in this, you are finding out what about that protein? The first amino acid, exactly. So, yes, the N-terminus, the first amino acid. Edmonds reagent tells you whatever the first amino acid is in a peptide, yes?
For those types of questions where you have like the whole sequence, for the answer choices, are they always like N to C or can they be N to C or C to N?
If it's not labeled, they're always written N to C. If they're not labeled, you always assume that it's written nitrogen, as in the first amino acid, the nitrogen terminus is on the left, and the carboxyl terminus is on the right. Okay. So, one thing to know is Edmonds reagent tells you the first amino acid, right? And you go look at this amino acid here. This is only the side chain for it, and once you learn your side chains, you'll recognize that this is the side chain for tyrosine. I will give you a bit of a clue here. This notation, C6H5, is a shorthand notation for a phenyl group. So I'm looking at a phenyl group with an alcohol attached to it. That is just tyrosine. Okay. So that's how the clue for Edmonds reagent is given to you. They draw it like this, and you're just supposed to identify the side chain. Yes?
What does PCH mean?
I have no idea. It's the structure for the Edmonds reagent. It's like a really complicated looking structure. I don't know what it looks like off the top of my head, but it's just the shorthand notation for Edmonds reagent. Okay. The other thing you should know, and it's technically given to you in a table, but you should know this fact about it, is what carboxypeptidase tells you. It's the opposite of Edmonds. Edmonds tells you the very first amino acid. Carboxypeptidase tells you the very last amino acid. If you want to know which amino acid it tries to look for, you go look in the table right here. You look in the table right here. I thought it was the next teacher here. Carboxypeptidase A tells you that it's going to try to look for every amino acid except for proline, arginine, and lysine. What this means is that carboxypeptidase A in a peptide, if it finds any amino acid except for proline, arginine, and lysine, it'll rip it off if it's at the very end. Carboxypeptidase B is only going to look for arginine and lysine at the very end. And carboxypeptidase C is going to look for all of them if they're at the end. Okay? The reason why I give you that is because sometimes there's a clue that will tell you this. Let me highlight it. Whoops, the result is no reaction. Is that carboxypeptidase A and B are mixed up and applied to some unknown peptide, but no reaction happens. What can you conclude? You go down to the table and you see carboxypeptidase A looks for 17 amino acids. Carboxypeptidase B looks for 2 of the ones that carboxypeptidase A could not do. The only one they can't cover is proline. So when the clue says you use these 2 things, but nothing happened, you can conclude what amino acid is at the very end of the sequence. Proline, right. Question, yes?
Can you just memorize that?
I would not suggest. I mean, hey, if you hear it enough, maybe you can memorize it. But this is given in the table. This information here is given in the table. Yeah, you don't have to memorize the enzymes. Is that what you're? If that's what you're asking about?
Carboxyl A and B, and there's no reaction. I can conclude that.
Yeah, it'll always be like that. It won't be any other way. Yeah. Okay.
Because I know that in SI, you actually solve it.
Yeah, you will have an example of this. So whatever way you want to memorize it, if you want to know why it is that reason. If you just want to memorize it, you can memorize it also. It's whatever you prefer. Okay, those are 2 facts there. Yes?
What if it just says carboxypeptidase and it doesn't say anything?
They'll have to say one or the other. Yeah, and if it, in the very unlikely chance they don't tell you which carboxypeptidase is used, you can assume all of them. So in other words, you pretend that the carboxypeptidase looks for all the amino acids. Yeah.
What's CBR?
CNBR is cyanogen bromide. It is a chemical, and that's the next part that I'm going to go into. It's a chemical that cleaves the C side of methionine. Cyanogen bromide cleaves the C side of methionine. So, after you know the thing about Edmonds, after you know the thing about carboxypeptidase, the next thing will be given to you in a table, but you should know the next fact, is that these lists of chemicals and enzymes have specific targets that they like to cut in an amino acid sequence. They only cut the C side of their target, which is a fancy way of saying they cut the right-hand side of that target. Okay, and we'll do an example in a second. So cyanogen bromide is going to look for every single methionine in an amino acid sequence and make a cut on the right side of it. Trypsin is going to look for every single arginine and every single lysine and cut the right side of that amino acid in a sequence. All right, so now that we're given that bit of introductory information, let's try one of these examples. Let me try this one, because this is a good mix of stuff. Let me highlight it. Okay, so we are given a pentapeptide that shows the composition of methionine, alanine, arginine, and proline, and we are given the following clues. Your extra credit question will be this style of question where they want you to sequence a pentapeptide. To get full credit on the extra credit question, you need to not only give the answer, but you have to write out your explanation for how you get it. I don't need 10 paragraphs, but I need for each one of these steps like a sentence describing how you knew proline was the last amino acid or something, right? And we'll get to that in a second. Yes?
So, like, we can't just write out, like, for, like, the mix of A and B good reaction, we can't just write out blank, 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 pro?
You need to do that at first, but you need to write like a short statement saying why you know to do that. Like, and it is the following, okay?
Before like that
Yeah.
A and B, can we say that because A and B yielded no reaction with proline?
Yeah, as long as you write it out. But you should write, and excuse me, you should write why you know it's proline and nothing else. And you say carboxypeptidase A looks for everything except for those things. Carboxypeptidase B only looks for two things. Left over is proline. Okay, so let's try to solve this real quick. All right, pentapeptide with these amino acids. So the very first thing you can write down. Get rid of the highlight here. Or you know what? I'll do it down here. Okay. The very first thing you can write down, and I won't write the sentence explanation, you'll have to include that on your own, is that your pentapeptide means that you have five possible amino acids hooked together. Those 5 amino acids have to be some combination of methionine, alanine, arginine, and proline, because that was what was given to us. Because I've only given you 4 amino acids, but there are 5 spots, you need to know that one of them, we don't know which one yet, is going to show up twice, correct? So, that's the first thing you can write down. The next thing you can write down, we already explained it, so I won't spend too much time on this. We find a clue that says a mix of carboxypeptidase A and B resulted in no reaction. We already know that that's a fancy way of telling us that the last amino acid is proline, correct? You can go review your notes on why that is. We just spent time talking about it. The next clue tells us that trypsin resulted in an arginine, a free-floating arginine, and a tripeptide. That is not a typo. We'll explain what's going on in a second. The clue is telling us that trypsin was used and it cut its target, and the result is, in your solution floating around, is some amount of arginine and a tripeptide. So I don't remember what trypsin cuts. We're going to go down to the table, or is it up to the table? Go up to the table. Trypsin cuts the C side of arginine and lysine if they are present in your pentapeptide. So we go look at this. We don't have any lysine to worry about, so I don't care about lysine.
Would you give us one that has growth pepper?
Actually, yes, it is possible that that can show up, and that's fine. You will have enough clues to eventually figure out which one is which, but yes, that can happen.
Which one cuts stuff on the end side of the
Technically, and I don't like explaining this because it is more complicated than this, technically only carboxypeptidase does because it removes the last amino acid, but don't memorize it like that. Memorize it that everything on that list cuts the C side of its target, and don't worry about anything else. Yes?
Since it says trypsin resulted in arginine and a tripeptide, does that mean that there's two arginines?
That's exactly right, and that's the next thing to get at is the clue tells us, after using trypsin, that we get arginine and a tripeptide. But if you add that up, that sounds like there's only 4 amino acids in here. The reason why that is that when you actually do this in the lab, there's no way to tell how many arginines you have. So all this clue is reporting to you is someone did this experiment and they say, I detect arginine and a tripeptide. Based off the previous information, that must mean there must be 2 arginines somewhere in this thing. There must be 2 arginines because the only other thing floating around in the solution is a tripeptide.
And they have to be next to each other, right?
And that's the next part to figure out is we have 4 possible options to put 2 arginines. There is only one arrangement that we can put the 2 arginines for the clue to make sense. And feel free to guess until you figure out the right places. And you're right, they have to be next to each other. In fact, the 2 arginines need to be the first 2 spots, and we will see why in a second. When you do this enough, it may be easier to guess right away where certain things go, but feel free to guess where to put certain amino acids and then check to see if you guessed correctly. So I'm going to guess that I put 2 arginines here in the first and second position, and I'm going to use trypsin on them. Trypsin targets the C side of arginine, so it's going to cut the right side of this one and make a cut, and it's going to cut the right side of this one and make a cut. We are left with floating arginine in solution, floating arginine in solution, and a tripeptide. We put the arginines in the correct spots. In fact, if you try any other places to put arginine, it will not give you arginine and a tripeptide. Okay? So feel free to guess until you get the right answer there. And I highly suggest you check your guesses as you go through. All right? So we've so far figured out our sequence is arginine, XX, proline. Almost done. Cyanogen bromide resulted in a tripeptide and a dipeptide. You go look in the table, it'll tell you that cyanogen bromide cuts the C side of methionine. So this information is telling me where methionine should go. One second. Methionine should go in either position 3 or position 4. Anyone want to guess where it should go?
3.
Position 3. So let me include it real quick. If it goes in position 3, let's check that our guess was correct. Cyanogen bromide cuts the C side of this thing, and we end up with a tripeptide and a dipeptide. So we put methionine in the correct spot. Yes?
Could it be possible that he'll give us a tripeptide and dipeptide, but the order is dipeptide and tripeptide?
Yeah, that's a good point that you bring up. It doesn't matter what the clue says. It doesn't matter when they say that they're given a tripeptide and a dipeptide. That doesn't automatically mean the first fragment is tripeptide and the second fragment is dipeptide. They're both floating around in solution. We don't know which one was first and which one was second. Yes.
So then how would you solve this problem?
Because if you try to put it in position 4, what do you end up doing?
Free.
If you end up putting it in position
Oh, okay,
4,
never mind. Because what I'm thinking is that sometimes on the problems I've done, it goes not N to C, it goes C to N, and that's why.
you don't have to worry about any C to N's. It'll always be N to C. Yes.
Besides just using logic, all we need to know is Edmonds tells us the first one,
Yep.
and carboxy
Tells the last one, yeah. And everything else is in a table.
Yeah, thank you.
Yep. All right, so there's no more clues, but there's one more spot. Process of elimination is the very last spot must go to what amino acid? Alanine, because that's the only one we have left. And that'll be your answer, okay? Again, short explanation for each one of these, and you'll get full credit on there. You have a question? Okay. All right. Okay. Let's see. There's plenty of other examples here. Feel free to try them. And I'm going to spend a long time typing up the answers to this stuff, so you'll see the answers when I'm done typing it up. Okay. Check the notes. Yes, it will be on Canvas when I am able to finish typing it up. All right, 2 more things. from chapter 5. Two more things from chapter 5. One of those things I actually just finished talking about at the SI session, which is recorded, so I'll post it on Canvas as well. This thing I talked about on Monday. So you may be given a question where the way they phrase it is that there's this brand new peptide that's 10 amino acids long. And they perform an experiment where they use some enzyme and it ends up cutting it into two pieces. Don't care about that enzyme. All